Hello, everyone. Welcome back to this virtual conference, our second annual water quality conference called Uncharted Waters, Equipping Sterile Processing with a Roadmap to Compliance. I am so excited that you've decided to join us this Friday. I know everyone has a busy schedule, and being here means you're dedicated to water quality issues, water quality compliance in your departments. And so for that, we thank you, and we're excited that you're here. A quick note related to continuing education credits and getting your certificate. For those of you tuned in live, at the end of the last session, you'll be automatically redirected to the CE survey. Once you fill out that survey, you'll be able to download your CE certificate. For those of you who will be watching the rest of the conference on demand, you'll still get an email at the end with a link to the CE survey. If all else fails, you can always visit beyondclean.net and click on free CEs to access that as well. I am so excited for this next session in this event sponsored by Verda Water Quality Systems. Our next speaker is John Prendergast, the Senior Decontamination Engineer at NHS Wales. John and his team provide validation services, advice, guidance to health boards and trusts within Wales in the United Kingdom, and work closely with the Welsh governmental departments to deliver policy and strategy in this unique and often complex field as we know it. John's presentation today will give a unique perspective on water use for decontamination of medical devices within the UK. Beyond Clean is dedicated and committed to bringing a global perspective at our virtual events. And so this presentation is certainly in line with that. John will talk about sterile processing departments across the world and how they tackle their water quality challenges. John, I'm so excited to welcome you to the virtual stage. Without further ado, take it away.
Many thanks, Lindsay, and good morning, good afternoon to everybody within the audience, whatever time of day it may be, wherever you live. It's a great honour for me to represent the UK in this delivery today, and I'd just like to thank Beyond Clean for what a fabulous job they're doing, highlighting the importance of decontamination sciences, not only in the US, but across the world as well. Okay. Oh, jump one slide, too many. So, yeah, we're going to look at why, what, and how we use water for decontamination. Predominantly based upon my experience in the UK, but certainly I welcome interaction from you as audience members with your experience across the world and in your own localities and regions. Okay, so my name is John Prendergast. I'm an authorising engineer decontamination. So what is an authorised engineer decontamination? Well, we're quite a unique band of brothers and sisters that are predominantly UK based and Ireland. So we are basically we're qualified engineers. We have some of us that are microbiologists, pharmaceutical experience as well. And we promote the highest standards of decontamination. We promote best practice based upon our experience, looking at equipment, facilities, utilities, etc. So quite a mixed bag of applications, and it's a very important role here in the UK. Disclaimer, obviously we've got to get this out the way. It's just the presentation I'm delivering is my personal interpretation for educational purposes only. It's not particularly related to my employment within the NHS, albeit it is based upon my experiences, the guidance illustrated as part of my employment, and the guidance and standards of the UK, Europe, and beyond with ISO requirements. Okay, so what are the objectives of the presentation? Well, I think I'd like to deliver a learning curve to explain how the UK has adopted rinse water for decontamination of medical devices. What emphasis we put on the legal requirements within the UK. We'll look at the various water sources that are available and how we engineer systems, we monitor, and we investigate the requirements to make sure our patients are safe, our devices are safe, and that we are achieving the highest standards possible. So why is water so important? Obviously, we all come across water every day of our lives. Water is life, basically, and our existence on Earth is dependent on water, or lack of it, as you may say. Civilization is built upon the use of water, and I think it's probably in the 19th century, as a population, we became aware of the importance of water, water quality, sanitization, and basically how important it is to not, because water, incorrectly treated or of a poor quality is a hazard to health, and it really is a bad product. So how does that link to our particular industry? Well, let's look at our industry. Decontamination, it's a challenge, okay? It's a combination of processes, including cleaning, disinfection, and/or sterilization, that we combine to render a reusable item safe for further use, okay? So as we all know, effective decontamination requires the attainment of acceptable standards at all stages of the decontamination life cycle. But I pause there. I'd like to ask you a question, or pause for thought. You think of it. What is our main weapon when we are undertaking decontamination? Think of it. Is it the human being? Is it the equipment? No, it's water, okay? We use water for every stage of the process. If we think of steam sterilization, we use water to generate the steam. Cleaning, this particular group is very clean. The emphasis is on cleaning. Without water, we're not going to clean. We use it to flush. We use it to rinse. We use it to irrigate. Every part of the decontamination activity uses water to achieve what we set out in the objectives. So if we think initially of our washer disinfectors, so why is water so important? Well, it dissolves cleaners and treatment agents, okay? It transmits mechanical forces and heat to the surface of the items that are being washed. It dissolves dirt and impurities. It flushes them away, sends them down a drain, okay? And if we think of the wash disinfect cycle, if we are using an A0 of 600, which is 90 degrees for 1 minute, how are we going to get that energy to the devices we are cleaning? Well, we're using water. We're using water to transmit that energy. Okay, so why is water so important? Well, unfavorable water can have an adverse effect both on the treatment, the appearance, and quality of the devices we are decontaminating. Okay, so the devices are made of high-grade stainless steel, other high-grade materials. They appear with polished surfaces. They've got passivation layers. We don't want to damage any part of those devices, okay? So when you think of your particular region, your locality, your country, what rules govern those localized conditions? What do you have to tell you? What water standard must you use? How do you make sure your washer disinfectors, they're using the correct water source? And what's all the fuss? Water is water. It's been on Earth since time began. Okay. However, we must realize that water should not reduce the effectiveness of any processed chemicals. We use wash detergents. Those detergents are manufactured to deliver a certain efficacy. if we have bad quality water upstream, it's going to reduce the effectiveness of those chemicals. It should not recontaminate the device. As we know, water is a vector for transmission. We don't want any impurities left on our device, okay? It needs to be of acceptable microbial purity. And if we think of our devices that we are actually using, the water quality must be compatible. Now, when we look at the likes of RO water later in the presentation, RO water can be quite aggressive. So we need to make sure our devices are man enough for the job. Okay, so where did it all start in the UK? Well, basically it all started with this document, which is Health Technical Memorandum 2030. Okay, there were guidance, there were standards before, but this really started the ball rolling in the UK. So what standards do we use for water supplies for decontamination? Health Technical Memorandum. We have, for any healthcare delivery setting within the UK, we have a HTM that is applicable for the activities, okay? Now, if you're a healthcare organization, you have to adhere to the HTM guidance or provide evidence. If you don't comply with the HTM, you have to provide evidence that you are complying to a better standard, okay? So if you think of the HTM, in the UK, we have what you call the highway code for driving, okay? If you adhere to the highway code, you cannot be prosecuted for poor driving. If you don't adhere to the highway code, you will be prosecuted. In healthcare, it's exactly the same thing. The HTM is the highway code, okay? So HTM, basically, the knowledge and the application of HTM 2030 began with the advent of well, basically mad cow disease, okay? The awareness of bovine spongiform encephalopathies in the UK, the risks associated with it, really set us off looking at decontamination standards. The life cycle, we looked at the life cycle, and prior to 1997, sterilization was king, okay? A lot of emphasis on sterilization. The hospital I worked at as an engineer, they had porous load sterilizers. They had what you call a tunnel washer, which was a washer disinfector with a conveyor. So in my experience, this conveyor was not validated. At the end of the shift, some of the larger members of staff would pull the devices through the conveyor. The instruments would not see any water. Well, with the advent of HTM 2030, that went away. Validated washer disinfectors were highly of the highest importance, and part of that is water quality. Okay, we learned that we can't sterilize, we can't. Prion proteins, etc., are known to show resistance to traditional steam sterilization methodologies. So we mention the inappropriate, the inadequate washer disinfectors. There was a national survey. Okay, so what happened? Members of the civil servants associated to UK government went around all healthcare facilities looking at their decontamination processes. That led to investment, okay, because they realized we need to invest in washer disinfectors. We need to look at our cleaning methodologies, because if we don't clean appropriately, we can't sterilize. So as identified, we had a change in focus. Cleaning, washing is critical, and water is an essential part of that process. Since HTM 2030, was in production. We've had revisions, and the current document that we use is HTM 0101. Now, in the regions of the UK, the devolved administrations, we have in Wales, we have the Welsh equivalent. Scotland, they have the Scottish equivalent. In Northern Ireland, they have the Northern Ireland addendum. Okay, so where I work, WHTM is adopted. Very similar. However, there are different interpretations for certain applications. So the HTM 0101 documents, primarily for decontamination, 0101 is dedicated to sterile services, management of surgical instruments in the acute care settings. HTM 0104 is dedicated to decontamination within laundries. HTM 0105, that's dedicated to the dental side of our industry. And then HTM 0106 is dedicated to flexible endoscopy. Okay, all very interesting documents. If anybody wants a bedtime read, I would certainly suggest this will send you to sleep. Okay, highly complex documents and very scientific, evidence-based, very good and very informative. So HTM 0101, segregated into 5 sections. Part D is dedicated to the washer disinfectors themselves. Okay, very much based upon new guidance, looking at the prion protein risk. It goes into residual proteins, how we monitor residual proteins. One thing I'm particularly keen of, it's okay monitoring protein left on devices, but we need to clean. We need to make sure that protein does not adhere to the instruments, okay? And part of that is effective cleaning. So in the HTM, there are dedicated water quality requirements for washer disinfector applications, okay? There are identified limits for each particular water quality parameter. It's very important to know what sort of water quality we have at source, okay? We may need to improve the water quality because we may have certain parameters that we don't want. And there will be a need to continually monitor the quality. We don't just commission equipment and water quality is as it is at point 1. We need to monitor at periodic frequencies, okay, to make sure the source is consistent, to make sure any purification system that we have installed, making sure it's working correctly. So here's an example of what is in HTM 0101 Part D. As you can see, the limits in there are quite precise. Okay. So where do we sample this water? Do we sample it at the purification plant? Do we sample it at source? Do we sample it at the washer disinfector? Well, ideally, you should have information of all parts of the process. Okay, we need to know what sort of water is coming into the premises. We need to know our purification plant. We need to know, is it doing what it should be doing, and we need to know the quality of the water at the washer disinfector. You know, we've got a washer disinfector is connected to a source supply by a flexible hose. Flexible hose are known to develop biofilms, so we need to make sure point of use. Okay, I think that's very important when you're collecting water samples. So if we look at the typical wash disinfector cycle configuration, okay, generally 5 stages. This can differ dependent on application, but generally flush to remove gross contamination, wash with detergent to remove any remaining soil, okay? What types of detergent do you use? That will differ. Is it the appropriate detergent, you know? And what should we do if we're using detergent? Because if you think, we validate these washer disinfectors, any changes in detergent could impact on the water quality we have at source, okay? We rinse to remove chemical additives. We disinfect. I mentioned earlier A0 of 600, which is a European figure that we actually use. Typically 90 degrees for 1 minute, okay? And then we dry. Highly important, we want a dry product prior to any packaging for sterilization. Okay, so should we have different water qualities at each stage? If we think of the pre-rinse, does that need to be the same as the final rinse application? Have we got consistent water in the UK? I know there are many of you from all over the world, different localities. Is your water quality the same as mine here today? What's your thoughts? Okay. It is going to be different. The UK is quite a small island, but we have different water qualities at each particular region. So when we go back to our HTM 0101 Part D, okay, so if we look at the flush application, yeah, cold potable supply is appropriate, okay? If we look at our cleaning using detergents or enzymatic cleaners, we need to have soft potable water. Softened water, it must not interact with any process chemicals we are actually using. If we think of our final rinse, obviously that's the final stage of the wash disinfect process. We need to have water of a high level of purity. And atypically we use systems called reverse osmosis for that stage of the process. It doesn't have to be RO, however, that does generally tend to be the more common method that we use across the UK. So HTM 2030, of which HTM 01 followed suit. What sort of parameters do we look for? What sort of parameters do we test? Well, we look at the appearance. We look at the pH. We look at the total dissolved solids in the supply. We look at the hardness. We look at the chloride content, conductivity, heavy metals, iron content, phosphate, silicate, TVCs, environmental mycobacteria, bacterial endotoxins. So, as you can see, there's a host of parameters that we need to monitor, to control, to make sure our surgical devices are safe. The appearance is not compromised. The device is not compromised, okay? So that's where we are. That's where we are in the UK. That's typically what we control as part of the rinse water application. So if we look at hardness, now I challenge you to, next time you're in a hotel, look at the state of the kettle, okay? If you look at this particular example I'm showing you, look at the scale formation around the element, okay? Is that healthy? What is that going to do to the element? And think, if you're making a coffee, you're making a cup of tea, you're drinking that, okay? I hope I haven't put you off drinking coffee in hotels, but just look at it the next time you're there, okay? So scale formation is a major no-no when it comes to decontamination. I've seen it in many washer disinfectors, and indeed I've lost many washer disinfectors before their end of life. So it's not cost effective, because a washer disinfector is an expensive piece of equipment. The last thing we want is scale to present within the internal chamber. We don't want it on the element. We don't want it on the devices, okay? So within our technical guidance, depending on application, we have limits that are below 50, below 200, okay? And we measure that routinely. Conductivity. Is conductivity a major risk? Well, not on its own, but conductivity is a very simple parameter to check. It's a very simple parameter that we can test, okay? We take a sample, we have a calibrated meter. We put the meter in to the sample, and we can check the ability to conduct an electrical charge, okay? And that gives us a reading. So if you think in the UK, we check the conductivity on a weekly basis. So we're checking our supply routinely. It's simple. If key properties of the water change, the conductivity will change, okay? So we know whether it's an RO plant, whether we got softening plant, we know something isn't right. And we know that gives us, we get our maintenance teams in, and we have a look what's going on upstream, okay? Bacterial endotoxins. Now, this is a very debatable area. Traditionally, when we presented our guidance, because it's based upon pharmacopoeia standards, bacterial endotoxins were seen as highly important. So what are endotoxins? Well, an endotoxin is part of the outer membrane of the cell wall of gram-negative bacteria, okay? They are lipopolysaccharides, okay? And such substances as E. coli, Salmonella, Pseudomonas, they all present with endotoxins, okay? But what are the consequences of endotoxins, okay? What harm does an endotoxin do to patients? Well, in the correct quantity, endotoxins cause toxic shock. They can, at worst case scenario, they can create a serious patient reaction. Ultimately, it can lead to death, okay? Now, in the guidance documents, we put in a standard of 0.25 endotoxin units per mil. Is that over the top? Is that extreme? Well, I'd probably say yes, okay? Controversially, I would say I think we've gone too far. So, are there other tests that you can do to measure endotoxins on instruments? Well, yes. You can measure, rather than measuring the amount of endotoxins in the rinse water, could you look what's present on the actual devices itself? And yes, you can. It's always a huge discussion point with microbiologists, the risks with endotoxins, okay? And I'm sure we could have a discussion on that today and again. But I think the primary importance I would like to deliver to yourself is endotoxins are important, and we do need to control. Okay, so total viable count. Again, a routine test, okay? Total viable count is a test that estimates the total number of microorganisms that are present in a water sample, okay? Now, there are defined limits, and endoscopy applications, the level of TVC is quite well controlled. But again, I ask, are TVCs an actual risk? Well, they are. Uncontrolled total viable count does present a risk to patients. However, is it more important we know what organisms are in our actual water sample, or are we just primarily concerned with volume of bacteria? I leave that to you. So, colonial morphology. This is identifying what what bacteria, what is actually in our supply. Now, to me, I think this is what we got to be asking the question. If we have one colony forming unit of Pseudomonas, does that present a greater risk than 50 colony forming units of other bacteria? Okay, it's all for discussion and based upon differing applications. Okay, so the other variables we look at, which we've touched on earlier, if you think of excessive chloride levels within rinse water, well, that is known to cause pitting and corrosion with surgical instrumentation. High levels of pH, high alkalinity, can damage devices. Silicates stain, create a yellowish, purple discoloration. If you are undertaking a surgical procedure and that device has discolored, what does that do to you as a surgeon? Are you happy? I'd say no. Heavy and non-ferrous metals, again, scaling, discoloration, evaporation residue, spotting and scaling. So you can see each particular parameter left uncontrolled will have an impact to our devices that we're actually using. And there's a couple of typical examples of device damage. Okay, if we look top left, that is foaming that was found in a washer disinfector as a result of uncontrolled parameters. The instruments at the bottom, you can see discoloration, damage created by poor water quality. So how do we know what we have? Okay, I ask you the question, do you know what water quality you're using in your decontamination centres? Do we know what are the risks, what are the applications, what are the consequences? Okay, so in the UK, if you look at the HDMs, we are quite precise. We do weekly testing, quarterly testing, we do annual testing. Each period has greater test requirements. Okay, the primary importance is to develop a trend. Okay, know what we have routinely, and know when we are when we're moving away from a validated position. Then, as I said earlier, we can do something about it. So we have all these tests, but how do we interpret the results? Okay, is the SSD technician qualified to interpret the results? What's your thoughts? Is the decontamination manager qualified to interpret the results? We need to engage with the correct professionals, okay, when we are interpreting the results. The user, the unit manager, highly important, yes, needs to be included. The authorizing engineer, decontamination, that's the likes of myself. Yeah, we need to involve those people. The microbiologist, they understand the consequences of colonial morphology, as I identified earlier. The decontamination lead, the infection prevention control lead, and the authorized person decontamination. The authorized person decontamination is the site engineer that is usually responsible for items of equipment such as the RO plant. Okay, so all these people need to be involved when we are interpreting the results. That diagram on the left is a typical example of trend analysis. Okay, the results we have on the right are being scrutinized, and we put them into a graph to show what we are developing during the test period. Because remember, not all samples are, as they say, we can have corruption when we take the samples. We can have corruption when we deliver to the laboratory. Okay, many things to actually consider. Now, a big thing I'm keen on is audit. It's okay having these results delivered, but we need to make sure, look at our results, let's review them, Check they're compliant with our local policies, our national guidance. Ensure there are no anomalies, okay? We have to have routine audit frequencies, okay? Ensure we've touched upon the relevant professionals. Make sure people are trained, okay? Aseptic sampling, handling of samples, okay? Highly important. Logistics. Do we need to control, especially if we're doing microbiological testing, do we need to make sure that our samples are adequately cooled as we get them to the laboratory? Okay, some of these laboratories may be 50, 100 miles away. What effects will keeping them in a warm container have to that sample? I'll leave you to think about that. Some of you live in warm climates, a lot warmer than the UK. Does that give you a problem? Yeah? I asked the question, okay? And we have the results delivered. Do we retain those results? Do we keep them accurately, okay? So in a year, in 2 years' time, if we had a clinical incident, can we go and look at those records, okay? I'll leave that with you. So if we think of what we're doing in the UK, are we too stringent? Are we going over the top? When we look at what you're doing in the US, in various other countries, do you have similar systems, or do you take a more lenient approach? I'll leave you to think on that one. So, personally, my interpretation of the HTM 2030 and the revisions, yeah, I honestly think water for injection possibly is going over the top. However, we needed to have an acceptable range of variables, okay? And that give us something to work from. Some of the limits documented will have no effect on patients. However, they can have impact on the quality of the devices, as shown, okay? We need to control our rinse water, okay? How do we manage that? And if we think of particular systems, what are the governing factors choosing such systems? So we now come on to the final part of the presentation. What influences the standard of supply? Okay. So if we look at the UK, a typical graph supplied. If we look at where the water quality or where the water source originates from. If you look at London, which is the Thames, in comparison to Northumbria, top of the map, which is Newcastle area, north of England, basically. Okay, so if you look, Thames, London, majority of the water is from groundwater sources. Now compare that to Northumbria. The higher percentage is from upland reservoirs and streams. Okay, so is that going to give us a different product? Well, certainly in the UK it does, okay? And I'm sure in parts of the US and other parts of the world, you are in identical scenarios. So, the source water supply is crucial, okay, when we're determining types of water treatment. However, think of our internal distribution system, okay? Where is our reprocessing centre? Where is our decontamination centre? Is it in the middle of the hospital? Is it on the outskirts of the hospital? Or are we in a central source? Are we in an industrial centre miles away from any healthcare facilities? Does that impact on our choice of water? If we think of a large hospital, the internal distribution system is huge, okay? Are those pipes all maintained? Are there any dead legs present? Have we got biofilm formation? Yes, we will have, okay? I guarantee. What sort of devices are we processing, and what are the instructions for use, okay? Any of you been involved with robotic surgery, okay? You look at the instructions for those particular applications, it's a can of worms. It gives us major problems. They will ask for things that probably the standard decontamination instructions will not ask for. Types of device we're using. What are the risks? You know, are we going into highly sterile areas of the body, or are we just piercing the outer membranes of the skin? Does that make a difference when it comes to water quality? And probably the most important factor is there a terminal sterilization process to follow? If we think of our friends, the flexible endoscope, generally there is no terminal sterilization. But in sterile service units, generally they go into a terminal process. So are the risks any different? Do we need this high quality water? So atypically, types of water treatment used within the UK. Well, if we think of potable supply, so the water regulators within the UK require that water has to be delivered to a certain standard, okay? So basically you have drinking water quality. So is that safe? We have mechanical purification systems, of which, as mentioned earlier, RO is probably the most common. We have high-level filtration, okay? We have UV technology. We have water softening systems. We have chemical dosing systems. Atypically, hospitals, if we're talking about drinking water, will have chlorine dioxide dosing systems at source, where it enters the healthcare facility. We have distillation plant. I bet if you look at your system, it's a combination of the above. It's a combination of a number of treatment systems together, okay? So if we look at the water softener, what are we trying to do with the water softener, okay? So we've highlighted we've got hard water upstream, okay? So we need to reduce that hardness. I've shown you the impact of hard water, so we need to make sure that's controlled. So we have what we call a softening plant, okay, a water softener. And the basic principle, we are replacing calcium and magnesium with sodium ions, okay? If you look at the drawing there, okay, we have a mass of very small resin beads, and these resin beads are negatively charged, okay? So these beads absorb and bind positively charged calcium and magnesium ions, and in the process release sodium, okay? So that brings our hardness levels down. Now, there's a lot can go wrong with a water softener if it's not maintained correctly. And the number of audits I've gone to, if you look at the brine tank on the left, okay, that brine tank has to be checked and updated on a daily basis, okay? So we need to add solution to that to make sure the softener is working correctly every day. Crucial part is to get your maintenance team on board, okay? Because if we don't, you're not going to have a softener working as required, okay? Hard water in, softened water out. We've got filters, we've got a number of things there that need controlling, and maintenance is critical, because if you don't maintain this water softening plant correctly, it can become more of a source of contamination than if we don't use it at all. RO, okay, reverse osmosis. So we're now using pressure, okay, pressure greater than atmospheric to apply to the surface of a membrane, okay? So we're forcing impurities through the membrane, the impurities go to waste, and hopefully upstream then we've got a purified water quality. Now, an effective RO plant is considered to remove up to 99% of impurities. However, again, maintenance is critical, okay? If we don't maintain our membrane, if we don't maintain other critical components, this RO plant can give you more headaches than sometimes the worth, okay? That's the line diagram. Pressure, we can contaminants to the left. We force the water through the membrane, okay? The impurities are rejected, and we've got permeate water upstream. Now, if you think of the world at the moment, and I think we all get sustainability, we all get effective use of resources and utilities. This is quite a utility-heavy process, okay? We waste quite a considerable amount of water. So when you've got your RO plant, you need to consider, can we reclaim that water, okay? Something to ask your estates, your engineering teams. So filtration, similar sort of technology to RO, only probably lower specification. We're trying to achieve exactly the same intention, okay? A filter media which can have a number of gradients. If you think of screens, they're in place to remove very coarse particles. Strainers probably react up to 20 microns, filters below 20 microns, okay? Make sure if you've got a filtration system, you have it rated appropriately. Now, what sort of impact do you think filter blockages can have? I talked about the downside of an RO plant. Are these any different? Certainly not. If you've got a filtration plant, you have to make sure you commit to change these filters routinely. Monitor the performance of the filters, okay? If you look at that particular photograph on the left, that's a filtration cascade. We've also got a UV capability on that, on the outgoing side. So look at the gauges, okay? The gauges are measuring the differential pressure across the filter housing. So as it becomes blocked, those gauges will give a higher reading, okay? Should be recording those readings on a daily or weekly basis, okay? Depending on your own localized policy. So there we are, untreated water in, it goes through the filter, and we have filtered water coming out. Any sort of contaminants, suspended solids accumulate on the filter material. Hence, it's very important to monitor our filter performance, okay? So if you look at a purification system, you can probably look, it's a combined system, okay? If you look bottom left, feed water in, pre-filter. Then we have a pump, RO plant, okay? Water quality sensor, sanitization ports all the way along, okay? Then we have a reservoir. We have a ring main, a loop. Why do we think the ring main is important? Why are we circulating the water, okay? You look at the ring main, we have a UV lamp. We have another filter, okay? During that ring main, you're going to have connections for washer disinfectors and any other equipment you may have connected. So, if we come back to our HDM documents, do we think they need to be developed around RO? Is RO the gospel that we need to follow? Is it necessary for all applications? Look back on what we've discussed today. What's your thoughts? I'll leave that with you. The important factors of a water supply. System design, okay? It should always minimize the potential for biofilm in line with local supply, okay? Do we think the material of the pipework is important? If you think in healthcare today, cost is becoming a major factor. We have plastic compositions. Should we be having plastic compositions, or should we stay with traditional copper or stainless steel on our distribution systems? Again, I'll leave that with you. Maintenance, highly important, okay? If you have any water treatment system, it has to be maintained correctly. And should it be maintained in alignment with manufacturers' recommendations? Or should it be maintained in alignment with local policies, local problems? Okay, probably a combination of both, because the manufacturers will give you generic guidance. Okay, you will know your local difficulties, your local problems. Consistency is a crucial part of the specification. Okay, we have to have a consistent supply. So we're coming to the end now. The essential requirements. Purification for localized conditions. Okay, always remember that. I think in the UK we've gone too much down the way you have to have RO. Look at your local supply. It's a multidisciplinary decision-making process, okay? As the SSD manager, it's not you in isolation. You use the other people within your organization to help, to assist, to guide. System maintenance and monitoring, important. We talked about testing. We talked about maintenance. Training. If we think of our sampling techniques, let's train, let's understand, let's know why we are taking the samples and what the variables are, what are the important factors. We prevent stagnation, we audit, we look at the results, we assist. Crucially, should be no Pseudomonas and there should be no Legionella. If you think of a water distribution system, two basic thoughts. If you've got hot water, keep the water hot. Okay, you don't want it reaching tepid temperatures. And if we have cold water, you keep it cold. Does this present a problem? It shouldn't. So the water quality standards within the UK, the HTM provides a good starting point. Okay, I would recommend that people use local policies to determine the specified limits as satisfactory. There are genuine risks associated with TVCs, endotoxins, and dissolved chemicals, okay? But the risk depends on the type of the device, the type of the procedure. Do we need better evidence for some of the test limits? Well, yes, it's got to, like everything else, it's got to be an evidence-based interpretation. Some of the HTM levels, yeah, they can probably be adjusted without any clinical instrument or machine implications. We may not need RO. RO is expensive, capital investment, and running costs. However, we may well need it as well. It's based upon local interpretations. Testing, monitoring, all the way along, highly important. So, just to sum up, I'd like to thank you for listening. And are there any questions?
All right. Thank you so much, John, for that presentation and for shining a spotlight on decontamination practices and water quality in the UK. We do have a couple of questions that have come through for you. The first of which says, hello, if we use critical water for TIR 34, is it okay for the UK?
Oh, well, I can't say I'm totally familiar with that document, so I don't know whether you can help me, Lindsay.
You know, I bet other subject matter experts on the panels this afternoon certainly can, so we can address that question in later sessions for sure. Yeah, this next question says, what is a closed loop on an RO system?
Well, a closed loop is basically it's as it says, really. It's a closed circulation system, so it wouldn't be open to atmosphere. Its distribution is distributing from the RO plant all the way along, and distributing at the required points.
Okay, excellent. Is there any one takeaway, if you were to sit down with someone who hadn't come to this particular session, what would your primary takeaway for them be?
Well, I think guidance is important. We need to make sure we are all working to a set standard. We can have our little local interpretations, but the UK is very, very standards orientated, and I think those standards are based upon a good background. You're thinking of the pharmacopoeia standards, and that gives us a great evidence-based basic template to come from. So yeah, without that, without standards, without guidance, where does that leave us? Probably anarchy.
Yeah, absolutely. I appreciate that sentiment and completely agree with you. A couple folks wrote in saying, great presentation, John. So thank you so much for sharing your insights with us today. It's been a pleasure to have you on this Beyond Clean virtual event. We're excited to talk more about global standards and about global practices at future events this year through Beyond Clean. So John, I'm sure you'll be back in one way or another. We'd love to have you. I was talking with one of my colleagues about how soothing your voice is, nonetheless. So it's just great to listen to you as well.
Yeah, no, it's a pleasure to join you all today.
Excellent. For those of you who have additional questions for John, you can find his contact information on the right-hand side of your screen in the speaker bio tool. Certainly connect with him on LinkedIn. Feel free to email any specific questions that you have for him related to the UK. For those of you who are jumping in halfway through the event, we are going to transition into our additional 3 sessions that are all panel discussions. So you won't be seeing many slides this afternoon. We'll be having some panel discussions related to water quality. So we're really excited for that. My colleagues Bobby Parker and Hank Balch will be joining me for those sessions. If you do need a break, we will be taking a 10-minute break between this session and the next. Feel free to do what you need to do before joining us for the second half of this water quality conference. We're excited that you decided to spend your Friday with us. For those of you who have sent in requests wondering where to get these songs that are playing between sessions, Beyond Clean released a sterile processing album called Beyond the Tunes last year on September 1st, International Beyond Clean Day. And if you're curious what that full playlist entails, simply visit our YouTube channel and search for Beyond the Tunes. So thank you for those of you who have shown such enthusiasm about these songs. John, thanks again for joining us today. We're excited to learn from you more at future events, and we look forward to talking with you soon.
Thank you very much, Lindsay. Thank you very much, everybody. Have a great weekend.
All right, everyone, keep your browser window open. Stay tuned for the next session. We'll see you in about 10 minutes.